for some reason you have no voice. But here I am again. Let's try this one more time. Hello. I am Joe the Science Bro, and we are doing Science Talk again. I believe we're on episode 3 with Dr. Balin. He is graciously lending us more of his time, mostly because I need more time to plan the rest of the schedule because it's difficult tracking people down. I want to take a few moments just to thank Dr. Balin for being very gracious with all of his time and allow me once more to welcome you. Dr. Balin, thank you for joining us today.
Well, thank you again, Joe. It's my pleasure. Happy to be here.
Well, I know that this has been a very interesting experiment so far, and I think that this has gone very well for my channel, my audience. I know that they've been enjoying it. I've been enjoying it, and I hope that you've been enjoying it as well.
Oh, I certainly have been.
Great. I am very happy to hear that. So, we've been talking the past couple of weeks about some of the origins of science, some of the things that are currently going on. Last week we were talking about being a scientist in general, but one thing that we haven't really discussed lately is the publishing aspect. We kind of touched on it a little bit last time, but this time I kind of wanted to get into it just a little bit more. Tell us how publishing works.
Okay.
Tell us, try and guide us through the quagmire of peer review and publishing in a journal, because for me, it makes sense because I've done it before.
Yes.
But for most people, they haven't. So walk us through it.
Okay. So typically, with scientific publishing, you have data that you now have gathered in your laboratory, and now you've written it into a scientific article. A scientific article typically involves having an abstract, having an introduction, materials and methods, results, and then a discussion. And you have to have references there too of all this work that you've done, reference other work that has been done previously that has led you to your work. Okay, so those are the components of a scientific paper. And then once you have the components there, and even before you have the paper finished, often you're looking into the literature as to what is the appropriate journal in which to put your information. So for instance, if you're doing something with neuroscience, you could go to quite a few different journals that publish on a neuroscience theme. Often the journals give you information about the particular types of work they would like to publish. For instance, if it was like molecular neurobiology, they would want you to publish on something, molecular techniques that you've used to understand a problem of neurobiology. So then you would take your paper and have write it up. You would then have a cover letter, and you could then send this off to the journal, and you could do that online. We used to do it by snail mail.
By mail, yeah, or by fax.
Or by fax, that's correct. So now you could do this online, you submit it to the journal. Once it's submitted to the journal, it's given a number, so it's like coded, and then the journal and the editors from the journal decide where and to whom they want to send the paper for review. And typically the reviewers are 2 to 3 individuals that are expert in the field in which you're publishing. And the article is sent to them, or actually they are invited to actually review your article. And they can either accept or decline. If they accept the review, then they have about 2 to 3 weeks in which to review this article and critique it. And the critiques then are written up by those reviewers, sent back to the editor of the journal. And in the critiques, they could say, this is a perfect paper, we will accept outright. Or they could say, no, the paper, we think it's really good, but it needs some level of revision. And it could be minor revisions or major revisions. And those revisions could involve anywhere from small items of misspelling or some syntax errors that are there in the paper, to confusion as to how the data is presented. And they may ask for a better representation of your data, or they may ask for a better explanation of the data that would be put into the discussion, for instance. Or they even may ask you to even consider doing an additional experiment to further confirm what the information is that you now want to publish. So you have a variety of different types of critiques that could come back. And based on those 2 or 3 critiques from those 2 or 3 reviewers, you now have to modify your paper, which we call a manuscript, and now justify what changes you've made and how you've met the reviews or the critiques that were now sent to you, how you've accomplished actually meeting those critiques. And so you're basically now saying, okay, I can do another experiment, or I can't do another experiment because we believe this is, enough information to actually tell the story that you're telling in this particular manuscript. So we look at manuscripts as you have a story to be told with information that's being now supplied, and a discussion about that is actually now telling a story about what it is you've done and what did you find and what it means.
So, is there any possibility in the process that, ego gets involved and these reviewers who, for all intents and purposes, are relatively faceless in their review process, and to a certain degree they kind of should be, because we don't want it to be interpreted as a personal attack, but is there any possibility that someone may be jealous of the work that you've done and might attempt to sabotage it via just an unending list of revisions that you want from it?
There are cases where the numbers of revisions that are being asked for are to an extreme. We usually don't see that with reviews, but every now and then there could be a reviewer that is thinks that you should have a more complete story, or one that would be even expanded beyond what you've already done. And sometimes those reviewers, what they're asking you to do, it's really now beyond the scope of the real reduction or reduced problem that you've been studying. And you can actually now basically tell the editor of the journal, write back to the editor and let the editor know that you think that this particular review is going too far with what the information is that you already have. And in order to obtain more information, it may require months and months of additional work that you believe would be a more complete story, but one that is outside the scope of what you're trying now to it's like saying you have a short story or a novelette versus a novel.
Okay.
And you're now trying to get it across that this is enough information to get into the scientific and public realm to help understand a particular problem. Now, you can continue gathering more and more and more information ad infinitum, potentially, and never get a publication out there. So you have to know that you have to stop at some point. The way many of us look at it is that your work is a lifetime of work, and it becomes a tome over time. To have a single article as its own individual tome and to be 100% complete in a story is usually not what occurs. You're usually building on information, adding to the information with your findings, and now you're building your novel, for instance. So these are like chapters of a novel, and you may be doing one chapter at a time. It's still important to get that information out to the other scientists in the community so that they can take that information, analyze it, and if they're doing something similar, use it to build upon their own story.
So in the regard that you're referring to, it sounds as though the editor in this case is almost like a referee, making sure that,
Yes.
or a moderator, or someone who's an observer of what is going on, and basically saying what is fair and what isn't fair in the review process from what you're saying.
That's correct. And many times you'll have 3 reviewers, and 2 reviewers are very close in their reviews, and one is an outlier, which is very interesting. Usually, if you have 2 out of 3 that are pretty close with their reviews, the editors will side on their side and agree with those 2 reviewers of whatever they suggest to be done with revisions of your manuscript. And then if they decide that, okay, you have now taken your manuscript and you've revised it, the editor then can say, okay, it appears that you've revised it to the level that was asked, and we will now accept your manuscript.
So it doesn't go to it doesn't go to a second review process?
No, it doesn't. So you just have that one review. Often, once you've gotten it back and you've made those revisions. Now, if you haven't revised it to satisfaction of the editor, it could be sent back to you again and said, no, you need to even revise it more to meet the critiques of the reviewers.
Gotcha.
Okay, so that could happen. Typically, that would be a more extensive review, and that would be whether or not you've really met the criticisms by revising your paper accordingly. But if you have an acceptance of that manuscript, it now goes to copy editors at the journal site, and they now put it in a format that is appropriate for the journal. And once they have it in that format, they will send you a preprint so that you now can look at the preprint of this, of how it will look when it goes into the journal and then is published within the journal, or published online. That would be in accord with the journal. And you can have publishing online and as well as hard copy publishing as well. So then when that occurs, you have the opportunity, usually it's about a 48-hour turnaround, where you can correct minor things in your article. You cannot add content or any significant content. You may be able to add a word or two, correct misspellings, maybe correct a reference if it's now noted that the reference wasn't actually put into the preprint properly. And then you can then send that back. Typically, we will get a PDF of that, so you can send the PDF back with your corrections to the journal. They make those corrections, and then it's ready for publication.
Have you ever been in the editing chair or the editor's chair before? I know that some scientists out there do take on that role.
Yes. So I have one journal that I'm an associate editor for.
Okay.
And I've also then edited, guest edited a number of different in a number of different journals. So as an associate editor, I would be sent different manuscripts that have been sent out for review, or that I would even review myself to see if it's appropriate for the journal.
Right.
So I've reviewed many different types of manuscripts based on my expertise and what's been sent to me. But as a guest editor, for instance, it's typically of a dedicated volume of work that has been asked for. So it would be on a topic, a theme and a topic that I'm familiar with. And then I would actually now suggest different people to be contacted to actually invite them to write manuscripts to be sent into the journal to be put under that topic for that particular volume. And I've done that a number of times as well. And typically after that happens, you get the people to submit manuscripts, you then get reviewers for those manuscripts, and once those reviews are done, manuscripts accepted, typically you have a volume that those that go into. Let's say it was a topic of infection and Alzheimer's disease, of which I work on. These papers would go under that topic, and then whoever the guest editors are, and sometimes you have multiple guest editors, you would write an editorial based on what those manuscripts now are talking about. And you would now give an overview or a summary of what these manuscripts really are focused on and what they mean for the field in general.
So, how has the journal experience changed from when you first began your scientific career to today? Other than the assistance of the internet, how has it evolved? So I remember, looking at older articles and seeing them being much shorter than they are today. Has there been some kind of a change that has required more expansive discussion of the narrative?
Well, it's interesting because with all the different techniques now that you can use to address a question in science, a problem, to solve it, sometimes you can use one technique and publish on one technique. Sometimes you may have 2, 3, 4 techniques that you're using in which to publish something. So here you need to get data based on your techniques you're using. So you could have a biochemical analysis, a immunohistochemical analysis. You could have a molecular analysis like a PCR, an RT-PCR. You could have animals involved. So depending on the experimental paradigm that you're working with and those experiments you're using, you're now collecting data that is taking the story and filling in with multiple techniques different aspects of a story. Typically that takes a longer time to get the data, but also it leads to a larger manuscript, a more complete manuscript, a larger a write-up of that work.
Okay. So, tell me about conferences. So, is a scientific conference something that anybody can go to?
In essence, yes. Anybody could attend a scientific conference. You would have to register for that. There's a registration fee. And once you've done that, you would get access to the different information that is actually being presented at a scientific conference. So if you're in person, for instance, you could register online or register at the conference itself and then attend the different sessions. In the different sessions, you typically have what are called platform sessions or oral sessions, where somebody is giving an oral talk. Sometimes it's a keynote speech, which is now longer than the typical speeches at a conference. So it could be a keynote speech you would see in a symposium where there's multiple talks on the same theme. You have different sessions typically, where the sessions then, you could have mini symposia where you have shorter talks on the same theme. For instance, they may be 15-minute talks, but you might have 5 of those based on the theme that you're talking about. So anybody can attend those. You can sit in the audience, you can listen to this, you can ask questions at the end. There's a question and answer period as well. And this is how you gain knowledge about that topic. Then you also have typically what we call poster sessions. And the poster sessions are typically where you're using like about a 6x8 board, in which you have now a large poster pinned up to it. And the poster basically follows the idea of a manuscript. So you'd have an abstract, an introduction, materials and methods, results, and a discussion. And you're basically in poster format showing a representation of what the work is that you've done. And often individuals will take those posters and use that information to write up a manuscript. So at the scientific conference, you can go and view these posters, and often you'll have the different researchers who actually were doing that work. At different points in time, they'll stand next to those posters, so they're there to answer questions. And that's usually like a very finite point in time, though. They may stand there for an hour, depending on how many people come up to talk to them.
Or how bad my feet hurt.
That's correct. So at times, if it's a really hot topic, you may have people surrounding that poster, talking to that presenter of the poster for a large period of time. Typically poster sessions run about 4 hours. So it's usually 8 to 12 in the morning and 1 to 5 in the afternoon.
Yeah.
Separated that way. And the posters that are in the morning are taken down during lunchtime, and new posters put up for the 1 to 5 session. So you have now 2 sets of posters in the same area of the convention center. Okay, on the board, on going up on those boards. And we have typically booklets or online information about what those posters are, the main topic of the poster, and the authors. So you can go searching for your topics of interest and go to those particular posters to talk to those presenters. And sometimes I've been at posters of my own at times where I've stood there for the 4 hours talking to people coming up. It never stopped. It was a continuous flow of individuals.
And then you've probably had those poster sessions where you're just standing there and it's crickets.
That's correct. That's correct. Again, depending on the topic and the interest in the topic at that point in time.
Yeah.
Yeah.
How different would you say it is now, given that we now have in the development pipeline, at least in the educational system, we have scientific writers that are now coming up in the pipeline. How is that going to, change or potentially even reinvigorate how we actually write and present our work?
Well, some people use scientific writers to formalize how the write-up would look when they take the data and they're now writing it into that scientific article. So that is something that is done. Usually that's a fee for service. That typically is done more in the clinical realm than it is in the basic science realm. I've never had people write up my work. I've always written up my own work. My collaborators and myself, we write up our own work, and we feel we would rather do that with explaining our own work than handing it off to someone who doesn't really know and is just taking the data and writing about the data.
Gotcha.
So that's one issue. The other is that in this day and age, because we have now artificial intelligence, we have, and some people have already started using this, is the artificial intelligence websites to actually write up essays and write up different articles using artificial intelligence. And that's now where it's extracting information and writing up like an essay or some type of theme paper based on how the information extracted from online sources, but it now is through an artificial intelligence website.
As the computer programmer in the room, I don't like that at all.
I don't either, actually, because
I'm not a fan at all.
And okay, so as an example, I have a colleague who actually looked into this and had an essay written using an artificial website, artificial intelligence website, and it wrote out the article, and it looked like somebody had written this. It could have been a scientist writing this, and it had references in it. And I tried to find the references. I literally looked in that essay and tried to find the references that were there, and could not, which is troubling to me, because now I don't know if the references were real or if they're taking something out of context and adding a reference to it and applying it to the topic in which the essay was written, for which the essay was written. That's a problem.
Depending on how the algorithm for the AI program was constructed, it is either doing one of two things. It is either working off of hello, Aru, good to see you. It's either working off of a database where it is actually taking specific pieces of each entry and then formulating a new reference, or it is grabbing from a database that exists and says, okay, this is related to this topic, I'm going to shove this in. So that's one of the reasons why I don't like it.
Yeah.
Because again, and this is why, if anyone ever asks really a lot of computer programmers, specifically the people that are doing like neural networks and AI work, yes, AI can simulate human patterns and human emotion, but they can't generate new. They can only work off of what has already existed or what is programmed into it. Even if it's self-writing,
Yes.
it can't create brand new, given the current world that we live in terms of computer programming.
Yes.
That is a limitation. So the difference between a computer program and a neural network and an AI. A neural network is kind of like an AI, but not exactly. It's kind of like the next step after a neural network. Is a program works off of a specific loop that once it meets its conditions, it's done. A neural network is a similar way, but it's used more for computational work. It's to simulate how the human brain would function given this set of criteria. And an AI is supposed to be a forever continuing loop constantly. But the thing is if you never give it a next input, it won't do anything. It'll just sit in its idle state unless you give it something to do during that idle state. So while artificial intelligence is artificial intelligence as we term it, it's not really what the term would suggest that it is.
Okay. I think I understand that.
Yeah, good luck. I wish I understood that. That's one of the major reasons why I don't like it, is because a lot of those algorithms are held behind closed doors because they're considered proprietary, they're considered IP. Why should I share my algorithm with you when you're just going to take it? And that's something that we deal with in the scientific community, because there are researchers out there that develop new techniques and they don't want them stolen.
Sure. Well, and one of the fears that I have with it is that, and it goes to not being able to find the references,
Yep.
is this material now being curated in a way that is taking bits and pieces of information,
Right.
putting them together, but putting them together in a way that no one actually has done before. And so is it really now through experimentation that you're seeing this issue, or is it through taking disparate information and using it in a composite to suggest that this has been done?
Right.
And that goes to the issue of when you see articles generated this way, you may not know it's been generated through AI
Yeah.
versus another researcher out there in the environment.
And I mean that kind of goes to what I'm saying. I mean it's not brand new information.
Right.
And there's actually current curated databases out there for scientific knowledge that anyone is able to actually access. So you have NCBI, which is the National Center for Bioinformatic Information. You have Uniprot and Swissprot, which are the European version of
Sure.
NCBI. And you have another one that is out of Japan and Southeast Asia, which is DDBJ, and that's a DNA database of Japan.
Okay. And you have PubMed, Medline.
Well, actually, PubMed is affiliated with NCBI.
Right.
So, at the end, I have to give some context of history for this.
Yeah.
And I hope you'll let me be the teacher in this regard.
Please.
At the end of the Genome Project, there was so much information that the researchers a part of that knew that it was too much information to just be held by them. So when they gave their report in, towards the end of it, I should say, and when they gave in their report to NIH and NLM, what they said was, you need to make a database. You need to make a centralized place where any researcher is going to be able to get the information that we have here and make conclusions of it. Because if you just limit it to us, you're not going to make much progress with this.
Okay.
And because as we both know, that project, as it got towards the end of its time, its funding and its personnel started to dwindle. And they knew that the only way that they would be able to make the strides that they did at the early portion of the project would be to have way more personnel, way more eyes looking on the project. This kind of goes to the one thing that we were talking about last week, where there was actually a Facebook game that was developed that was actually to have people recombinate DNA but it wasn't using a computer system to do it. It was using people, and it was using a puzzle-solving mechanism. And people were actually generating different ways that this DNA could be recombined. So NCBI formed, and they are a division of NSF, NIH, and NLM. And basically all they're there for is to just be a repository. Mm-hmm. They are a centralized repository. We use it for BLAST, which is a local alignment search tool. It's to let us find DNA and other genetic sequences rather quickly. But it's also got PubMed. It's got PubMed Central. And so the difference between PubMed and PubMed Central is one is curated, one isn't. That doesn't mean that the information in it is not valid.
Right.
I have to stress that because I've had a number of students in my time say, well, I only want to pay attention to PubMed Central because it's curated. And it's like, well, that's great, but the stuff in PubMed includes everything else.
Right.
The stuff in PubMed Central is, the best way to describe it is, one is a verified entry, the other is too new to have been verified. So when NCBI started really getting going, one of the questions that was raised was what about the people that are in like CERN and Edinburgh, because Edinburgh at the time was a large genetic center. What about the people that are in Switzerland? What about the people that are everywhere else? And they reached out to, I believe the company at the time was referred to as Expacy, and they said, can we kind of share information with each other? And the funny thing is that the European group was already doing the same exact thing with the Southeast Asian group. So the three groups got together and said, okay, we're basically doing the same thing here. Let's exchange information on a regular format, and they actually update each other as time goes on. So if you look at a what we call a GenBank article, which will look horridly evil to any person who has no idea what is in a GenBank article. It's like looking up a entry in the Encyclopedia Britannica, but it's written in ancient Greek. You will have absolutely no idea what's going on. You might be able to pick out a word here or there, but you won't really know what all that information is. And there's a ton of information there.
Oh, yeah.
So in a GenBank article, you'll also see a UniProt or an XPC ID as well as a GenBank ID. Bioinformatics as a whole has only come about now because of the Genome Project. But we do have a question. Okay. Who is doing the curating? Could they be leaving out information that they don't know is important? So the way that it was explained to me was that it is a third-party independent group. And it's, when I say a group, it's a very large group of the geneticists, bioinformaticians, and proteomists, which basically means it's a bunch of people who generate DNA information, generate the analysis of said information, and people who generate information of proteins and RNA. And they're looking at all of this, and what they're saying is they're looking at what's been referenced by this and what has been verified of this. So in a way, there is a possibility that they could be leaving out information, but because that's all this group does on a day-to-day basis, it's very unlikely. And because there's just so many eyes looking at it, because again, they update each other.
Sure.
So it's not just one area that's actually got whole control over the entire knowledge base. It's several groups. And actually, NCBI has several nodes of information around the U.S. that they actually store portions of their database. So no one location has the entire bioinformatic information database of the United States.
That's probably a good idea.
It's a very good idea. So, the reason why I went on the tirade is because my background is in bioinformatics, which is a little more, it's kind of like data science inside of biology. So I'm the new kid on the block. So there could be stuff left out that's just too new that hasn't been well, it's not that it hasn't been peer reviewed. It has been peer reviewed because in order to have an entry logged into GenBank, you need to have a paper reference that references that specific, what it's called an accession number, which is just a ID number. For those of us that remember card catalogs, it's the number that was up in the top left-hand corner.
Yeah.
God, that's going back a ways. Whew. I hate dating myself like that.
I know. You seem awfully young to remember card catalogs.
Hey, when I was in freshman year, I had to go to the journal card catalog. So here we go. How does verification and peer review differ? So peer review is what Dr. Balin was talking about earlier. It's the journal aspect. So that's when you submit a written, and please correct me if I'm wrong, you submit a written write-up of your work telling basically a story of what you did, and then your peers, your fellow scientists, read that after being given it by a journal's editor, and then they make comments, concerns, and questions regarding that information. And once you satisfy that, it then gets submitted. It's then published.
Right.
Verification means that it has been displayed by other people, so it's been replicated, but also it's been cited by other scientists. So, and Dr. Balin can elucidate this a little bit better because you've had this happen to you because you planted your flag, and then what was it? Harvard came about and basically did the exact same thing.
Yes, and it validated and verified,
right?
Yes, what we had done, and others did the same thing. Along the way, what happens with science is that when you do experimentation, we sort of talked about this last time as well, I think, that you can have others that repeat your work or don't repeat your work.
Yeah.
And you want others to be able to repeat what you've done so that what you've done is not something that is so anomalous that is unrepeatable. And that's the nature of science, is that we want others to be able to do that. So when you publish, you're actually telling others how to do the work as well by your materials and methods.
Yep.
And if they use the similar materials and methods, they should be able to follow along and use that as their basis for doing their experiments, and should get similar data that you've gotten.
And there's also a responsibility and a requirement of one of the members of the research project and the publishing group to be what's referred to as the corresponding author.
Yeah.
And it's their job to kind of be there to answer any questions of someone who is like, I'm repeating your work, and I have a concern because I'm not receiving the same result that you are. Can you tell me more in depth about what you did? And I think I was contacted by one old PI, which is primary investigator, that I worked for because I was the one who was doing the work. So he was like, hey, can you just give me, I know it was a long time. Can you remember anything out of the ordinary? I don't know if you've had to do anything like that.
I actually have. I've been contacted by others who have read articles of ours and contacted me about if we've done even additional experiments or if the paradigm, the experimental setup, if there were idiosyncrasies to that, anywhere from a pH issue to a temperature issue to a timing issue as far as how long you let the experiment run, etc. You have many, many different variables to consider. And you're hoping that when people try to replicate the work, they're really doing it in a very, very meticulous way, so that they're not skipping steps or they're not taking anything for granted that, oh yeah, I can do this at room temperature, I don't have to do it at 37 degrees centigrade or whatever. Right. So there can be those subtleties that can lead you down a path that you can not repeat what's been done. Sometimes you can repeat by changing even the experimental paradigm, which actually strengthens the argument of your data being very valid. So there are a lot of different ways that science has done that way, and the replication process occurs.
And another thing is that science, this is really going to get me into hot water. Science is not an exact science. it is very much an art form in that I could teach anybody how to do certain techniques, but there are certain nuances to doing those techniques. Like, it could be something as simple as the way that I'm holding the pipette could change the outcome down the line. And it could also be and I know this sounds really weird, but it has actually happened to me a couple of times, where even the altitude that the experiment is conducted at can affect the result, the efficiency, the end result. I actually had a colleague who did his master's work in the University of Phoenix, and when he tried to replicate it up here, he had an extreme issue, and it was because of the amount of humidity that was in the air.
Right. Exactly.
And it was something that he didn't consider because he was in Phoenix, Arizona, where it is dry.
Yeah. I've had the issue with humidity even here in Philadelphia, where we were doing immuno-EM experiments, and immuno-EM is using now antibodies that have a conjugate of a gold particle on them, and you're now interacting a tissue section with those antibodies to bind to a particular protein. And if you don't have the right humidity, your binding affinity and avidity actually can change, so that the antibodies don't bind as well one day versus the next day. And so you'd have to repeat the experiments many, many different times in that environment to sort of verify that you could actually get this to work, that it actually does work, and you'd have to take it to the electron microscope to see your outcome with this. So it led to a very meticulous, labor-intensive process. But I determined that, it was because of the humidity in the room that was changing the dynamic of your interaction between an antibody and an antigen.
Sometimes, and we're getting kind of a little more into the nuance of scientific research. So that's actually a good question. So, one of our members of the chat, he was using a neti pot, but he didn't boil the water properly.
Okay.
And it was well water, and he actually was afraid that he had gotten one of the what is it called? A brain-eating bug? The amoeba?
The Naegleria fowleri.
I think so, yeah.
Is the organism.
And
yes.
I told him, very unlikely, unless he's in a center of that particular amoeboid. And even then, still, very unlikely, because the United States water system is even if it's well water, it's still very unlikely.
It is unlikely. In fact, that amoeba will grow in stagnant, warm water, typically. Often in the southern states, they run into this. And the people that are contracting it have been swimming in a stagnant pond, a stagnant lake. Water's really warm, the amoeba is there, they've inhaled it while swimming, and it does go through olfaction into the brain, leading to severe amoeboid meningitis, which is often fatal. In fact, it's fatal in about 95% of the cases, unfortunately.
But like I was saying to him, it's kind of very unlikely that he would have gotten it. Even though we call it well water these days, it's not really true well water like it used to be, where it would just sit there.
True.
Don't forgive us. It's kind of a topic that we're not really used to hearing around this area.
That's true, but it's interesting though you bring up neti pots in general though, because of flushing through the nasal passages and trying to get off any viruses and bacteria, potentially fungal spores, things of that nature that are and amoeba that could be now accessing our the upper respiratory system that way, because it is respiratory as well as now olfaction in the upper part of the nose. So, but neti pots can work, and they can lead to clearing of material that way, so that you have a cleaner
intranasal do it too
environment.
often, though. You shouldn't do it too often, because again, we talk about commensal bacteria, which is
Right.
the bacteria that is already working with your system. And this is more of a recent development that we, and again, when I say we, I'm talking about the greater scientific community. We've recently discovered that the human body isn't just the human body, but it's actually a complex symbiotic system of multiple bacteria, some of which, when you take them out of the human body, we would identify them as harmful to the human body, but for some reason they live in concert with us. And actually, in the terms of your gut, you wouldn't be able to get the nutrition that you do without them.
Right. That's correct.
And
there are trillions.
Yeah, there's trillions.
It just in the gut, but also it goes to the issue of as you're saying, Joe, the microbiomes. Okay, so our bodies basically are a great petri dish, and we have microbiomes throughout. In fact, we have a microbiome in the respiratory tract as well. So and in our skin, in our gastrointestinal tract. It's thought that just about any accessible area in the body could have a microbiome that inaccessible to the environment, in essence.
Yeah, I mean it's very similar to the bacteria that actually live and grow around plant roots, the nitrogen fixators. Yeah. So there's a specific type of bacteria that are referred to as nitrogen fixators, and you actually need those in order to have your plants grow properly. If you don't, you'll actually see them grow very inefficiently because they can't get enough nitrogen. That's actually one of the big things that lets them grow so big. For those of us that remember the early 90s and everyone was miracle growing the hell out of the your plants, that's what you were doing. You were basically dumping almost straight nitrogen into the ground. It was nitrogen, I believe it was potassium as well. And that's where you would get the burned roots because it was too much nitrogen and potassium and phosphorus for the roots to be able to fixate properly.
Yeah, it's very true. And with us as humans, in our guts, the bacteria often can lead to the production of neurotransmitters, hormones, other ingredients that we need actually and are used by our eukaryotic cells for functionality. Sometimes even it can lead to disease, a leaky gut. You can lead to inflammation, systemic inflammation. That can also have an effect systemically, including into the brain of individuals. That's why a gut microbiome and brain interface is often talked about in this day and age, of how you could have that interaction influencing normal cognition or abnormal cognition at times as well, based on how your gut is functioning. So
it's very interesting.
It's a big issue.
Especially because, and again, I'm a bioinformatician, but I also spend a lot of my time in the bacterial world because bacteria, it's not that they evolve faster than us, they go through more generations than we do in a faster fashion. So they're very good for assessing inheritance, which is what I based a lot of my early schooling on. But bacteria are actually more sensitive to changes in the environment than we are. So it's actually a good thing that we actually have these microbiota because they do actually help. Wait, you mean our diet is important? Crazy talk. Believe it or not, and this is one of the debates that's currently going around is, some of the hormones that are given to certain cows,
livestock,
to certain livestock, the concentration of lime that's, given to, my wife is getting very into raising chickens. So one of the things that I've learned is that adding lime to their dust bath that they do, similar to like a chinchilla, can actually help them because it gets rid of mites. But if you use too much, apparently it can get into the meat.
Oh boy!
So it's there. I've always said that the human body teeters on this very thin line of balance, which we term homeostasis. But your body does this tightrope walk.
It does.
It really does this tightrope walk every single day, and all it takes is for just one thing to go haywire, and the whole thing's screwed up.
Yeah, so true.
We're talking about chlamydia pneumoniae because we're working on a project on it right now, and one of the theories that I have is that the organism is going to multiple locations, and that's because our immune systems are very good, very well tuned to a single infectious agent in a single location. But the moment that it starts moving out and branching to different locations, it gets overtaxed, it becomes inefficient, it doesn't know how to properly mobilize its immune response. So for a very well-engineered system, it's really bad at what it does sometimes. And it can actually make the situation worse at times too. That's why we have autoimmune disorders. Being constantly on antibiotics. Oh, I don't know if that's the greatest news, but there's probably a reason for that. So tell me about how things have changed with antibiotics in your research, per se. Because I know that you've kind of told me once or twice that antibiotics have been therapeutics in the Alzheimer's world every once in a while. How has that kind of changed, and why did you come to that conclusion?
Well, in the Alzheimer world, what's interesting is when we got into the infection area with Alzheimer's disease and realized there's an association of different types of infecting organisms with Alzheimer's, one of the issues came about was, well, has this been ongoing? And what about in people that had Alzheimer's disease and had another type of infection, like a urinary tract infection? And anecdotally, and this is from many, many anecdotes, of individuals with a loved one who was diagnosed with Alzheimer's disease, had developed a urinary tract infection, was put on antibiotics for the urinary tract infection, and became then lucid cognitively. And it was short-term and short-lived, the lucidity, but it could have been for days or weeks. There were some cases of a few months, and then they went and reverted back to a cognitive deficit. But that told us that antibiotics may actually be helpful in actually helping an Alzheimer individual. Now, when we found infectious agents in brain tissues of people who died from Alzheimer's disease, we were thinking, well, potentially those same individuals, if they had a urinary tract infection and went on antibiotics, potentially some of those antibiotics were helping by clearing or treating for the infection in brain tissue as well. And that we don't know, but we think that could help explain why they would become lucid even for a short period of time. So one of the theories we have is that chronic infections in the nervous system, and it could be bacteria as well, chronic bacterial infections, especially with the chlamydia organism, because Chlamydia pneumoniae is an intracellular obligate bacteria, meaning it has to be inside of our cells in order to grow. So it won't grow outside of our cells. So it acts more like a virus would. But because it's a bacterium, you could use antibiotics against it.
Yeah.
So one of the thoughts is that we could treat individuals with this infection with antibiotics, and they could potentially get better. Now, the problem with that is that you have to get antibiotics into the nervous system, and then you have to get them into the cells that have the infectious agent, and have a high enough dosing of those to kill the organism. And what we've realized over the years is that, unfortunately, this organism can actually develop a persistent form in cells, so that persistent form is like a spore, and it doesn't really get penetrated well by antibiotics, and or can become resistant to the antibiotics. So antibiotics would not be the only way to go about trying to correct the problem. Now, some people have tried using multiple and different types of antibiotics, and for other chlamydial types of diseases where chlamydia pneumonia has some type of respiratory problem, and have shown that you can actually diminish the organism to a point where you get better. Whether or not you've totally cleared the organism from your system is another question mark, though. And that's where it goes to the Alzheimer issue as well. Because even in Alzheimer's disease, if you don't totally clear the organism, it could become persistent and stay in the brain for decades even.
Right.
And we don't know for how long a person could be infected, and this could be an ongoing subtle process. And so the thinking is that if you have a subtle process or a chronic process in the nervous system, you could now subtly have damage that starts accumulating over time. And this may take decades, so that it's not until we're much older do we see the result of that. And unfortunately, that result could be neurocognitive change, neurodegeneration, Alzheimer's disease.
Wow.
There's a lot there.
No, there's a lot to unpack there. And I mean, this is one of the things that, you and I are dealing with whenever we're talking about this organism. And, the wider chlamydia community, they don't really look at pneumoniae as that big of a deal because, again, as you defined it, it requires a host cell, in order to grow and to be infectious. So why should I care about that when I have the STD form of it, which is trichomonas, to come in and basically wipe everything out? Why should I care? But you're kind of getting to that point. Trichomonas comes in, wipes everything out. The immune system recognizes that and responds. But with pneumonia, it can kind of hide away.
And yes, and also with the trichomonas organism, with the sexually transmitted disease, you get a level of hiding in the population because you get symptomatology, either asymptomatic individuals or very mild symptomatology. Unless, of course, you have other organisms on board, like gonorrhea, Neisseria gonorrhea, causing gonorrhea, or syphilis, Treponema pallidum, causing syphilis, HIV. These things will give you a more drastic and dramatic type of response in the body than chlamydia trachomatis does. And also with chlamydia pneumoniae, these things are more subtle organisms because they actually, once they've invaded our cells, they now have elicited a response by our cells, but that is not like a fulminant, meaning it's not like an aggressive response. It's sort of a subtle response, but it is a response and can be a chronic inflammatory response that now, once it's generated, it can keep going. Yeah. And if the organism is not cleared, either through antibiotic usage or through an immune process that we have, that in some people you can clear it, it will stay on board. If it stays on board, it can reactivate, it could grow in the right environment, it can create chronic inflammation. You'll get potentially chronic disease from that. With trichomonas as well, so the sexually transmitted form of chlamydia can also lead to chronic disease. And unfortunately, it's been involved with pelvic inflammatory disease and salpingitis and different changes in the female reproductive tract in particular, where it is even more damaging than in the male reproductive tract. In the male reproductive tract, we don't even know the extent that it actually may be causing significant damage. But there's some evidence that you can find it in the prostate at times. And is it an element of aging in older men with prostatitis? It's possible. In some cases, probable. Because once the organism is on board, it can become chronic because it also is infecting inside the cells.
So it's kind of topical that you bring it up, but did you recently hear, I actually just got the communication about it today, the HIV vaccine that recently came out, while it is safe, it's actually being shown to be ineffective in its most recent trial. If you could, could you shed some light on that trial and clinical trial process? I don't know if you've ever had to go through that process yourself.
Well, we really haven't run a drug clinical trial at this point in time. We would like to, actually, with some of the work we've found in the Alzheimer world with infection. But typically with clinical trials, the first part of that is to see that the approach with a vaccine or drug is not going to be harmful to the individual, so that it could be safely tolerated. And that even goes before there's any efficacy data that you generate with clinical trials. So the first level is to make sure that it's tolerable and you're not creating harm or adverse events and things of that nature. And this goes for almost any type of use of any type of drug. It could be a vaccine, it could be a drug, an antibiotic, anti-inflammatory, a chemotherapeutic drug, etc. And then goes to the next level where you're looking for a level of efficacy and safety issues, but efficacy, is there any level here that's recognizable, that it could be efficacious? And then once you find that out, that there seems to be some type of result or response, then you go to a large population with a phase 3, often phase 3, then to phase 4 possibly, where you're looking at large populations to see if you actually have any difference from the drug itself versus a placebo, which would be something that is not the drug, something that is innocuous that you would be using along in a blinded trial, to see if there's any changes between a placebo and the drug itself. If you don't see there's differences from placebo to drug, it would suggest that the drug is not really effective in a large population.
So, now that we're kind of talking about different things, different vaccines, different antibiotics, why do you think there's been such a major change? I mean, I have a theory as to why, but why do you think that there's been such a major change with the way that we're prescribing and we're using antibiotics, antivirals, anti-everything? Oh, wait, hold on. Has there been any data on, say, organ transplants affecting Alzheimer's? Specifically, if someone received a transplant from someone who had a higher risk of getting Alzheimer's?
That's a good question. I don't know specifics on any of the data that would show that. What's interesting there, though, with organ transplants, typically the individual would be put on immunosuppressive drugs fairly long term, just so you don't reject the transplant.
Almost
Immunosuppressive
completely.
drugs will actually affect your immune system and inflammatory processes in the body by diminishing them. So in some respects, that actually could help someone that has a risk for generating Alzheimer's disease by keeping inflammation at bay and keeping the immune system at bay, so that you're not responding to something that could be on board in Alzheimer's disease brain tissues, for instance, and or that you're blocking inflammation that's being generated early on, so you're not inflammation in the brain is not actually occurring, and to the extent that would lead to significant neurodegeneration.
So, anyway, going back to what we were saying, why do you think that there's been almost a pullback in the way that we've utilized antivirals, antibiotics, and most other treatment methods? It seems like there's been almost a complete change in the methodologies that we've utilized to treat certain things. I mean, I know from the DO perspective, because here at our institution we're focused on the DO perspective, which is to treat the causative problem and not just the symptomology. But in your opinion, in terms of science, why do you think there's been this pullback almost?
Well, part of it is that the use of antibiotics historically has been quite effective, but what it's led to, and I think it's more so with the animal population and what has gotten into our environment, into the water supply, as well as into the animal supply, what we're eating, as to increased antibiotic resistance, in particular with microbes, bacteria.
Yeah.
This can apply also to some level with antivirals as well. However, antivirals, you have less of that happening. You have much more with antibacterials. So they have mechanisms that allow for resistance to the antibiotic over time. And because bacteria multiply so rapidly, over time, genetically they're changing, so they're not affected by the antibiotics anymore.
Yep.
That antibiotic resistance has shown up with real serious diseases. For instance, with tuberculosis, there are now forms of tuberculosis because now you have a Mycobacterium tuberculi organism that shows extreme antibiotic resistance, meaning you can use some of the most potent antibiotics known to mankind now that are not effective against that organism, meaning basically you have a disease that is becoming untreatable with an antibiotic approach. The other part of this equation is that over time we have not kept up with finding and discovering new antibiotics to the extent we need. Now, some are still being discovered, and through chemical modification, even making some existing antibiotics more potent. So changing structures and things of that nature.
Adding cofactor drugs, which
correct. And so we are still behind the curve here. So the recommendations by many docs and the healthcare agencies, World Health, CDC, etc., is to minimize antibiotic usage until it's really absolutely necessary to sort of try to put a handle on and prevent extensive antibiotic resistance. And in some cases, there are organisms, even E. coli at times, that have shown up to be very resistant to certain antibiotics. Neisseria gonorrhea, which we mentioned for here you have a sexually transmitted disease. Some forms of Neisseria gonorrhea are extremely resistant to antibiotics. So in some cases, what it's come down to is some people believe using multiple antibiotics at the same time would lead to limitations on an organism becoming resistant to multiple ones at the same time, because the different antibiotics have different ways of acting. It could be something like acts against the cell wall of an organism. It could act on the RNA of an organism. It could act on the DNA of an organism. It could act on how an organism could actually replicate. But because over time you can have changes in the genetic structure of the organism and how things are made in the organism to resist the antibiotics, these effects of the existing antibiotics loses their effect and they're not working. So,
also, again, this is where, some of my colleagues and I kind of come in, is because we're now looking at the way that genes and proteins are changing and evolving and responding to different stimuli differently. We're not just synthesizing models to explain it. We're not just utilizing a computer to say, okay, let's simulate this. But we're also thinking of it in a different way. So we're no longer thinking of it as, okay, well, let's stop this process in the bacterium, and then that'll lead to its death. What we're actually looking at is, why is the bacterium becoming resistant towards this? And there's a couple of different things that exist that way, but one of the ones that sticks out in my mind, mostly because it affects penicillin, and penicillin-like drugs, is the beta-lactamase enzyme. And that's actually one of the things that I did my master's research with, was a neural network to identify the beta-lactamase enzyme. And what beta-lactamase does is it basically allows the bacterium to process the antibiotic, which basically means that they can chew it up and turn it into something that they can then utilize. Which normally it's kind of like if you were to ingest, it's the equivalent is, it's like if you were to ingest arsenic and then suddenly, instead of dying or becoming sick, you were perfectly fine. Your body was able to metabolize it in some way. And, this is one of the reasons why bacteria make such great models for genetics. And I don't want to say evolution, because evolution is such a hot button word these days, but for adaptation.
Adaptation, that's a good way.
Adaptation, I think, is a better word.
Yes.
Because in a way, that's what Darwin meant when he was referring to evolution, yes. But it's really adaptation. Someone's been building up a immunity to iocane powder. It's a Princess Bride reference.
Okay.
I don't know if you saw the movie.
Years ago.
Yes.
I don't remember
it's
that.
the drink that they drank. The one character says, "I've built up a resistance to iocane powder." It's funny that you bring that up, because there's actually a historical figure who spent his entire life trying to build resistance to poisons, and then when he tried to use poison to get away from his enemies, he had built up such a resistance that it didn't do anything. But then there's also the story of Napoleon, where he thought he was such a Superman that he took way over the dose that was necessary, and he ended up throwing up. But I mean, this is kind of the interesting fact. Yes, they were both poisoned.
Yeah.
So I mean, bacteria make such wonderful examples of adaptation because, again, it's like what I said earlier. They respond to changes in their environment so much quicker because they recognize it faster. Human beings, like earlier, I put this on because I'm sitting under the vent. And it's pretty cold at the moment, and I don't know why, because it's kind of cold outside. But
it's to keep us awake.
Yeah, it's to keep us awake. But it's something like that. I sat here for a little bit before, I put the this pullover on, but a bacterium would instantly change. The moment that it measured that issue, it would instantly change and adapt to it because that's how it survives. Unlike us, where we have several cells working in concert with each other, we're a single entity. You don't rely on me to survive, so we don't necessarily change that much.
And one of the ways bacteria does that too is by communicating with its neighbor.
Yes,
it can pick up other genetic material, extra genetic material, often called plasmids, that are the genetic material that allows for the bacteria to become resistant to the antibiotic. So it's actually allowing the bacteria now to increase its ability to actually make something that can, like the beta-lactamase enzyme, to actually combat now the use of an antibiotic in that regard. So the bacteria in our environments can pick up a lot of genetic material. But what I wanted to mention was, what's interesting is, and this is something that started, that I know of, in the 1930s in Russia, and is still ongoing today, even and now in the States and across the globe, is to look at how bacteria can be infected by what we call phages.
Oh yeah,
these
bacteriophage.
are bacteriophage or bacteriophage. These are viruses that actually can infect bacteria and lead to their death if it's a lytic phage, meaning it lyses the bacteria. And now it's in progress. A number of companies are working on trying to develop phages for specific bacteria to use them as killing agents therapeutically to actually combat different types of infections. And in theory, because they're very specific viruses for very specific bacteria, and each bacteria may have its own type of phage that could infect it, you could apply this technology to treating almost any bacterial infectious disease with its specific phage. So this is becoming an exciting time, and it's still not something that's used therapeutically at this point in time, but I think eventually it will be.
I'm just looking up something really quick because I want to make sure that I get this correct, because I always end up getting it wrong.
Okay.
I always end up getting the disease wrong. But I don't know if you've ever heard of the madness of King George.
Yes.
King George III. And I have a reason for bringing this up.
Okay.
So we had a question of, could there be a use for bacteria to detect illness in people, or is that something we already do? I go back to that because there was a way that they were utilizing to try and analyze his disorder, which is actually shown in the movie The Madness of King George III, and that's they were looking at his stool. They would, I forget what they called it the morning passage or whatever it was. And the reason why I bring that up is because of the gut microbiota involved. And one of the things that's noted, it's a good film, I do recommend it, the acting is very good, but they try their best to actually explain it as a disease, and not just a disease of the mind, but that it was something in his body that was affecting his mind, causing a temporary madness. They talk about how the color changes, how there's more water in it, which for you and I, that we have knowledge of the digestive system, there is a multitude that could affect color,
Yes.
could affect the makeup, and yes, I do understand that this is a very crappy topic.
Is that a comment?
Yeah. But if I remember correctly, they thought that it was porphyria.
Okay. Yeah.
And if I remember correctly, they believed that it was either a genetic disorder or a bacterium. But the thing is that no one knows for sure. Like for some diseases, when we look at it back in history, we can almost go, okay, it's most likely that.
Yeah.
Or it's most likely this. And if they had the knowledge that we have now, they would have decided that. But I just find it one of those medical and biological and scientific intricacies that we haven't figured it out yet.
Yeah. A couple things about porphyria. It was thought that that's where the idea of Dracula came from.
Yes.
Because of the drinking of blood.
Yep.
Okay, because these people become very anemic because porphyria, the porphyrin ring, is in the red blood cell around the hemoglobin. So you don't have the proper processing of hemoglobin, the red blood cells, etc. Become anemic and very pale, and also very subject. You cannot go out in the sunlight. It actually bothers you to be in sunlight. So it sort of goes to that of
I believe it's,
lore.
I believe it's you have a it's a melanin issue for that one, because I remember that porphyria is one of the side effects of genetic werewolfism.
Yeah.
But in that it's slightly different because it's a blockage of one or two of the binding sites of hemoglobin.
Yes. So off topic, but going back to phages, by
Yes.
the way. So I don't know if this is how this came about, but in the let's see. One of the earliest phages found was a phage, bacteriophage, that infected E. coli. And it's called the T4 phage.
I was about to say, is this the T4?
Okay. Guess what? The T4 phage looks like the lunar lander.
Yep.
Okay. If you remember what the lunar lander, the Eagle has landed on the moon, and probably it was designed after the phage that could had the legs to it to bind onto a bacterium to inject its genetic material into the bacterium.
I think it was. I know what you're talking about, and I remember reading the book on it. And I think what it was that one of the engineers was doing the math on it, and he found that this was going to be the best one to give them the most stable landing.
Yes, stable landing.
And I believe it was his cousin was a biologist and went, "I've seen that before. That's something that was just found out, and that's a bacteriophage." And he goes, "No, this is the lunar lander." And they had an argument about it because he was trying to say, "This is what I'm designing, but this is what I'm seeing."
Yeah.
So that's one of the few times. Well, if I say few times, but that's not true. It's one of the many times that mathematics and science have actually been doing something simultaneously, and they come about to the same answer together, even though they weren't working on it at the same time.
Yeah, it's interesting because I often think about how biology, sort of in a microscopic form, can also then be translated into a macroscopic form, we see this in biology a lot, especially with proteins and how proteins fold and how they twist and turn. And then we see like plants and how they grow, and trees and how they twist and turn, and even our own bodies are large, and other organisms. So there's like almost a mirroring from the microscopic all the way to the macroscopic of form. It's very interesting.
We have a fun fact, actually. The lunar lander was designed to be able to drop from 15 meters to the surface, but Armstrong put it down so super gently that they actually ran out of fuel on the way down.
I don't think I knew that.
I didn't know that. But then again, that's just goes to it. I mean, there are wonderful people with our colleagues at NASA, but that was just a completely different world there in the 60s. It was a completely different animal. I have a couple of colleagues that are what are they calling themselves these days? Space biologists or something? Or
Okay.
I don't know what they refer to themselves as, but they're basically just biologists that operate in zero gravity or minimal gravity.
That's interesting.
But it's a completely different world over there than what you would expect from the 60s, where it was just constant innovation.
Sure, and I still have a slide rule in my drawer here at work that most people don't know what it is when I pull it out. So, and that always reminds me of what they use for the calculations when they're going to the moon. And
never forget that a bunch of guys with
70s.
calculators and slide rules put a man on the moon.
Correct.
I can't tell you how many instructors I have that have told me that over and over again. I think even now in the space shuttles, they still prefer mechanical switches, meaning electrical input rather than fiber optic, which is what you would find today, because it has less of a chance of failing in different gravity, because it operates off of a specific signal. Electricity doesn't vary in varying gravities. It actually maintains the same. Same with a mechanical. The moon is as real as girls on the internet. Well, that's the funny part. I mean, I don't know about you, but I kind of get confronted with I've gotten confronted with conspiracy theories, which might be one of the major differences in our educations. I don't know when you were coming up, but mine was the age of the internet conspiracy, and I had to field every last question under the sun about vaccines causing autism, which they don't. Vaccines causing disease, which they don't. Well, they do, but they don't. It's hard to explain. We'll talk about that later. But also things like the moon landing was faked. The Earth isn't flat. Everything, and the Earth was proven to be round by an engineer in ancient Egypt.
Right. Good point. I would say the first conspiracy that I became aware of, and this goes back to the Kennedy assassination, and it's still thought to be a conspiracy to this day by many people.
Right.
Okay, even though the reports came out and they released a lot of information after 50 years of in the archives, and the Magruder film that showed the assassination, and then a lot of people that were witnessed ended up dying mysteriously. There's a lot there to unfold and unwrap. So that's one that I actually I don't dwell on a conspiracy there, but I think that there was more to the story than what we were made to believe.
Right.
Okay, so that was probably my first one, but in my age, and then throughout time, possibly because of that, and the level of secrecy behind it, led people to start thinking, oh, what are some other things that could be conspiracies? Now, I don't know before that the level of conspiracy that was being considered. But I do know after that, a lot of people then felt that there was more to a conspiracy issue than not.
So I touched on this briefly, and I don't want to get into it too much, because for you and I, we've whipped and beaten this like a dead horse, because every scientist has, and that's vaccines causing autism. And I have a reason for bringing this up.
Okay.
So for me, this is an extreme sore spot, because number one, it is an absolute fabrication of data that led to this. He admitted that he fabricated the data at one point because he was sued. And to this day, he goes around giving talks, even though his medical license has been revoked. But how did you respond to that? Because I believe at that time you were a researcher at that point. And how did you take, former Dr. Andrew Wakefield's report?
With skepticism up front about that, then with some, thinking about what is in the vaccine that's been given,
Yeah.
So we always go with, okay, what's the data? What does it show us?
Right.
And what's in the vaccine? Now, you have mercury compounds or something of that nature in the vaccine. Could mercury do something in the brain? Yeah, it could do something in the brain.
But not at that concentration.
But yes, exactly. So we're thinking, okay, did the vaccine cause autism? And that's led me to think differently about autism and infectious components for autism.
Right.
Okay, which gives you now a different view of how this could actually happen. And I don't believe the vaccine causes autism. However, I believe that vaccination can lead to more of a global response in the body that we still don't quite understand.
Well, that's true. I mean, you're stimulating the immune system
Correct.
in a way that is foreign to the body.
Correct. And so with that stimulation of the immune system, you get a lot of activity of different cell types. Also, that can occur in different regions of the body. So we think of it that, it's just the lymph nodes that are being activated, so you're activating T cells and then eventually B cells, etc.
The thalamus, isn't it?
Well, you definitely can activate.
Isn't that in B cell maturation or T cell maturation? I forget. It's been a long time since I've done immunology.
No, that would be the thymus.
Thank you, thymus.
Thymus.
Yes. Oh my God.
But and that, in young people, obviously it's very active. But in us, it's probably now dormant for the most part, unless someone develops a disease like myasthenia gravis, and then it becomes due to tumors of the thymus, thymomas, things of that nature, can be a factor. However, with vaccines, because they can activate the immune response. In young children, that activation of immune response, is to be protective against, in this case, it was measles, mumps, rubella.
Right.
The MMR vaccine. So here's a thought, is that in utero, you have now, during gestation, in utero, this developing fetus is in communication with the mother. We've now realized that there can be a lot of congenital crossover.
Right.
So meaning that what the mother has, antibodies, some infectious agents, etc., can cross into the fetus. And one of the theories and one of the ideas that I've thought of, and others have thought of this as well, is that potentially something is on board in certain now fetuses growing, then becomes a baby after birth. In this young developing being that has been on board that was contracted from the mother. An infectious agent. And there are probably a dozen different ones that can actually infect a fetus this way. And I lecture on this for the medical students, actually, congenital infections.
Oh, I didn't know that this was something so topical.
So yes, and one of them is on rubella, and I also do Zika, and I do listeria, and syphilis, and HIV, and cytomegalovirus. These things can be on board. Some of these can go unnoticed after birth.
Interesting.
Now, even into young childhood, they can go unnoticed. When is the MMR vaccine given? So
it's given 3 times, I believe.
Yeah, it is now. So it's given at a fairly young age. Could that vaccine lead to an immune response that actually has an effect against something that was already existing in that infant that was not recognized initially as being foreign,
This is very true.
in activating the immune system now is being recognized as foreign? It's just a question.
No, this is very true.
But that's a possibility
Yeah.
in some of these individuals that you had an effect that way that led to now damage in the brain, cellular change, etc., that we define as autism. It could be other diseases too. And we don't know enough about how that response could possibly take place and what are the other factors in individuals like that. So that goes to a different way of evaluating what did the vaccine do?
True.
Vaccine didn't cause autism.
Right.
Okay.
But it may have activated something that stimulated the response.
Correct.
But this goes to, the aspect that I was hinting to earlier. The human immune system is a wonderful, wonderful tool, but it can mess up very easily and actually can cause more damage to itself even, because certain types of allergies are autoimmune or mini autoimmune responses. But we have so many autoimmune disorders out there. Heck, lupus is still uncharacterized, and I think the most we know about it is that, or at least the most that we can agree on it, is that it's just an activation of the inflammatory response that is unchecked. But causatively, we don't know.
Yep.
I've heard everything from it's genetic to it's psychological to its diet to its exercise to it could be something of the phases of the moon, causing a tidal change that changes the humidity in the air, for all we know. And that, this is why I say to people, science is an ever-evolving thing. And that's both a good and a bad thing, because for us, we recognize that it's a long process that is ever-evolving and ever-changing. And I don't want to say normal people, because that's not fair, but for those of you that don't do research like we do every single day, that's a hard pill to swallow, because you want that hard answer. I mean, even to this day, I want that hard answer. When I read a journal article, one of the worst things that I can get from it is, so you're not really sure as to what's going on, but this is your theory. And sometimes that's all you've got.
Yes, it's true. And that's why collecting more and more data, studying the problem, evaluating the problem, and thinking outside the box often about the problem is actually helpful, because it gives you now a more well-rounded view of what the problem might actually be. By the way, I wanted to mention about the vaccines, two things. One is that it's been shown that people that have been vaccinated highly actually have a decreased risk for developing Alzheimer's disease. And we don't understand why. Okay. We don't understand why, but it has to do with the immune response and how that's working in more of a general sense in the body. And actually, it's been found and correlated with the BCG vaccine, the bacillus-calmette-guerin for TB.
Yeah.
Those individuals, and so you follow those populations, realize they have lower prevalence.
That's extremely
of
Alzheimer's.
So so there's a lower risk, and it's been shown a number of publications on that. So that's one thing. The other thing goes back to what you said about vaccines causing disease.
Right.
Okay, so some vaccines, and notorious for flu vaccines, causing Guillain-Barré syndrome, which is a peripheral nerve demyelinating process.
Yes.
That can be corrected. But what happens there is the antibodies that are made after the vaccine actually can interact with myelin that coat our nerve cells, our axons, peripheral axons, throughout our bodies. It can lead to paralysis. Some people actually are never cured from that. But often if you catch it early, if you do plasmapheresis, where you take out the antibodies from the blood, basically cleaning the blood from antibodies, the body will correct.
And again, this goes to what we touched on earlier. The human body has a very fine line that we call homeostasis, which is the operating conditions that you have, and because of that fine line. And the craziest part is, and this is, I still remember when they said that the Human Genome Project was now done and we were going to create personalized medicine for every person. I was like, that's great. How are you going to do it? But not every single person's immune system is the exact same.
Right.
You might require a certain level of viral load that will cause a certain level of symptoms, but for me it might be twice as much. For me it might be half as much.
Correct.
For me, I might just need to be in the presence of someone, and now I've got a full-blown bacterial infection.
Yeah.
This is one of the reasons why it's so good for people who are immunocompromised or on immunosuppressive therapies to constantly watch the CDC and guidelines from the CDC, because usually what's happening there is they're trying to get information out for those who are at risk
Yes.
for those things. And I know that, recently there's been a lot more of politicizing of those bodies, but, in the end, and again, this is because I'm an idealist, I do believe that those people are trying to do the greater good
Oh, yes.
in what they're doing.
I would agree with that too. Also, there's a journal they put out called Emerging Infectious Diseases that the CDC actually puts out, and it basically is, I believe it's a weekly journal of like what are the current emerging diseases.
It's the weekly reporting.
That are coming out across the globe. So it's a very
I believe
very
you can-
valuable source
I believe
of
you can
information.
subscribe to that almost through
yeah.
text message, and you can
I
get
think you
text
can.
message updates.
I think you can. Yeah.
I forget, but if I remember correctly, you can. Here we go. So I'll just read this in the way that it's written. So I recently saw a video. Showcasing a bionic extra thumb that was controlled by switches pressed by your toes. The interesting thing was that in brain scans, using the thumb was actually still activating the same area as the hand, not the foot, even though you're using your toes.
Interesting.
Yeah. Well, another thing that you have to understand is that all nerves in the body originate inside the brain.
Yeah.
There's no nerve bodies. There's no cell bodies outside of the central nervous system. So that actually...
Other than ganglia. Well, you do have ganglia.
Ganglia, I don't know. Are ganglia termed as neurons, though?
Yes, they're cell bodies.
Oh, that's true.
Yeah. And they sort of sit in a peripheral sense.
Right.
Okay, but then within the central nervous system, including the spinal cord, so you have all those nerve cell bodies there as well. So.
But it's so... I've seen the dissections.
Yes.
Oh, God, what are they? They're wood transfers, which is a type of printing method for images. And what they would do is they would actually dissect out the nerves, the axons that have stretched so long, and it's so interesting watching how long these nerves get. Also had several comments where people were discussing how easily our brains acclimate to different modes of interacting with the world. Well, that's the interesting thing about humans. We're the only organism that modifies its environment and no longer is modified by the environment.
That's a good point. That is a good point.
Yeah, no, I mean, I think it was as late as, and when I say late, I mean back in the past. I think it was as late as the 1800s was the last time that we were actually being modified by the environment, and that's the Little Ice Age.
Interesting.
Yeah,
it is interesting.
My dad was a history teacher for 20 years, so he was very big on those.
You
things.
pick something up.
Yep. I've experienced this myself, where I play VR games for a while and get so used to moving in the game that you unconsciously try and teleport while walking around the house, because the way movement is done in certain games is that you have a circle that just jumps in front of you, and you jump into that circle basically. So that's interesting. I know that there was a lot of videos going around the internet. There was this this game that would make you like walk out on a balance beam of a skyscraper while you were in VR, and all you'd see is people jumping, and they would literally jump in their homes. And there's so many videos of people just colliding with their TVs, walls, jumping down hallways.
No doubt.
But that's the sensitization of the brain. The brain acclimates to the environment that it's in. The human body is so interesting in the way that it adapts to certain sensory changes.
And we have a lot of senses.
Yeah.
So it's sort of like, and what we consciously are aware of versus what we're unconsciously aware of is different. And that's.
Well, I think
so much is coming in at
what is it? They
one
say
time.
we don't have 5 senses. It's 5 mechanical senses, and I think it's like almost 30 abstract senses. I think is what it is. I know that perception's one of them. But yeah, it's very weird.
It's weird.
It's weird because you're raised as a young kid. There's only 5 senses. And then you find out I get so dizzy after playing Beat Saber for too long, coming back to the real world. There's actually, I experienced this myself, but there's actually a phenomenon. I don't know if you remember the video games Guitar Hero or Rock Band.
Yeah.
But there was a phenomenon, an optical illusion, that the ground was actually rising
Oh, that's wild.
because things are coming towards you only in a specific area. So your brain just compensates for that one area, and optical illusions and everything as
yeah.
well. And there's one thing I did want to ask you about for Alzheimer's related.
Uh-huh.
What are your opinions on the music therapy aspect? Because I've seen a couple of videos where people who are completely unresponsive are given headphones, and I believe it was swing music was played for one individual, and he was very locked up,
Uh-huh.
and then he loosened up, his posture changed, and he almost started dancing to the music.
Yes. Well, I don't fully understand everything
Mm-hmm.
that's going on there, but I think that the music processing is also tapped into long-term processing and long-term memory.
Yeah.
And is because you still have an activation probably from that also into motor regions. So
Yeah.
And I say that because with Alzheimer's disease, we often think of it as more of the limbic system. The limbic system meaning an old system that is based on emotion, emotionality, a response, fight and flight response,
Yep.
learning, memory, personality, and long-term, and then going on into longer-term issues. However, the long-term memory issues are really preserved. Okay, so a lot of thought to be more frontal cortex preservation of those nerve cells and triggering those through different activities. Music being one of those, and I think because music in the brain is sort of connected into automatically remembering where you heard the music for the first time, like it's like smelling for the first time. Where did you first smell something?
How does so maybe you can elucidate into that? How does that trigger in us? I know that I'm getting into a great deal of nuance in terms of brain chemistry for memory, but how is it that we have such strong response, such strong and sometimes visceral responses? Because I've seen I've seen people who have been introduced to smells that were very close to almost odor to them, and they have an immediate response of expiation like that, where they're just vomiting instantly because they're reminded of something that triggered a trauma.
Yes.
How is it that actually works, or are we now hitting the edge of?
Well, our senses, it's interesting because many of our senses actually go through a region of the brain called the insular cortex, which is like it's right on the side of the brain, basically, but inside the side of the brain, of the temporal cortex, basically. But that region is still sort of now being discovered as to what it does. But what it's interesting is it takes a lot of the senses and puts them together and gives you the ability to respond certain ways. So, for instance, it's a region of the brain that if you came across something that you can't tolerate blood, you came across a scene where somebody was bleeding, you would start getting sick. Because you're processing now this information, it's going there, and it's now triggering other responses in the brain. So it's almost like this area that takes in multiple senses, processes, then gives you that response. With music, it may be a very similar thing too, that it's activating different areas of the brain so that it's the long-term memory, but also the motor regions of the brain. These are areas that are not as affected in Alzheimer's disease early on. So in mild to moderate, going into maybe close to severe, but not quite there, Alzheimer's disease, you would still have those abilities. If you go to severe Alzheimer's disease, you have so much damage in the brain. A lot of those people become non-ambulatory, incapable of functionality, basically. And so it depends on then the level and extent of damage in the brain. But we're still trying to figure out exactly how this works with auditory processing. We hear this, and auditory processing is tied into so many different regions of the brain as well. Just like all of our other senses, while we think of it, they go to one place, they go to multiple places. It's multiple activities that get activated.
Is that kind of like how we have certain preferences in regards to food, scents, sounds? It's to almost relive favorable memories. It makes me almost wonder if it's a evolutionary response to kind of force us to almost autopilot ourselves into more safe situations.
Well, that's a good point. Yeah, I guess you could look at it that way. And something that's more pleasing, a pleasurable situation, because the brain, there's so many chemicals there. Some of them are very pleasurable chemicals. The brain likes that. We, as translated to people, like to be not typically in pain unless it brings them pleasure, but it usually goes to a pleasure type of thing.
Right.
We like to be in that brings some level of homeostatic balance, I guess, is the way we could look at it to us.
Have you ever seen any link between blind or deaf people and Alzheimer's? Is it affected at all by the different wiring of the brain since they've adapted to the disability?
I don't know enough of the information about that. I actually don't. I haven't seen that. I still think those individuals can go on to get Alzheimer's disease, but I haven't seen really any major reports about that.
Well, it's kind of
that'd be an interesting way to look at it.
It's a microcosm of a microcosm.
Yeah,
in a way. So I mean that can be difficult. The only reason I know that is because one of the projects that I did was on chronic osteomyelitis, and we were looking for something that was occurring 1% of 1% of the time.
Yes.
So it is the proverbial needle in the haystack that you're trying to get at. But I mean, again, this is one of the reasons why I love having these talks with you, because some of these questions that we're getting
are great questions,
are extremely insightful, and I really do have to thank you guys. By God, is that the time? It's 6:21.
Oh boy. Okay.
Time flies when you're having fun.
Yes.
So just a few more questions, and then I think we'll end it for tonight. So
I can always come back.
Yes, that's true. You can always come back. Have you ever seen the film on Golden Pond?
Yes.
With I believe it's Henry Fonda
and Jane Fonda.
And Jane Fonda.
Yeah.
What is your opinion of the sequence in which they depict Henry Fonda's character's first Alzheimer's-like situation? I think it's the scene when he's in the forest and he just can't find his way home. Yeah. He knows that he has to go through the forest in order to get home, but he doesn't know where he's going, and it's just panic.
Yes. What's interesting there is that can be a symptom early as well as later in the disease process. Mm-hmm. So it could show up at different time points.
Right.
Some of that is due to damage in the hippocampal region with space and place cells. Mm-hmm. But also in the parietal cortex, where we are more processing our surround and where we are in our world.
Right.
And those areas are often affected, and I say often because it's hard to say 100% of the time in an individual, but often affected in Alzheimer's disease with a degenerative process. And so the cells are damaged. So what will happen there is that it can be noted quickly, okay, by being in either an unfamiliar environment or even in a familiar environment, but there's some difference about that environment at a point in time. Like all of a sudden you've encountered, in this forest scene, for instance, you've encountered the forest from a different direction than you typically have walked through.
You're off the path.
Right, you're off the path. And now you're going, oh crap, I don't know how to get back to the path because you're in an unfamiliar setting. I actually experienced this with an uncle of mine.
Okay.
And here's the description of this. Okay, this was up in Binghamton, New York, and I was out with my aunt and uncle. I was a kid. I was only about 12 or 13 at the time. We had gone out to dinner. We were heading back to where they lived. And he's lived in this city his entire life. He was about 72 years old at the time. And driving down the main road to go to his house, and there is now a fire, and fire trucks have blocked the road.
Oh.
And my aunt says to him, Leo, his name was Leonard, Uncle Leo, was, just go to the right. And he's going, well, I don't know how to do that. He could turn right, but he didn't know how he could go around where the fire truck was blocking the main road. And there was another back road to go around.
Right.
Okay. He did not know how to do that, even though he lived in this area his whole life.
Yep.
And we're going, is there something wrong here? Is he, experiencing, some type of an event? Thought at the time, even back then, is he in shock? What's happening?
Yeah.
And we realized that we finally directed him the way to go. Even I knew how to go from there. And I wasn't living in that town, just visiting. But it comes down to this, is that often that spatial orientation will be affected.
Yeah.
And it can at times be the first incident where you realize that this person is being affected, brain, or there's something wrong with the brain.
I've always characterized, mostly because my mother is very afraid of contracting Alzheimer's, because every member of her family never really got to long-term age, the elder members of her family. And there was also some adoption in the situation. So we really don't know all of the information regarding past family history, because, and again, it goes back to that generation of the 20s and 30s really didn't keep much in regards to records of that kind.
Right.
And it also wasn't characterized really well at that point. I don't remember. Please remind me, when was Alzheimer's characterized by Dr. Alzheimer?
About 1907.
Okay.
This was about when it was accepted that he had his first case. That
So it wasn't necessarily widely
Right.
Around until probably, probably the 30s. Was when it would start to be actively looked for at that point, because it would take a while for that information to disseminate.
Alzheimer was looking at that time, so was Oscar Fischer and Creutzfeldt-Jakob, leading to Creutzfeldt-Jakob disease.
I was about to say,
these were very severely demented individuals that were showing up, and they were following. And then eventually doing the autopsies on the brains and realizing they saw these structural abnormalities. So that was already ongoing at that point in time.
Right.
Prior to that, not so much. It was, a lot of people would just declare that it was arteriosclerosis, which was the hardening of the arteries type of process. Not even atherosclerosis, but arteriosclerosis. So it goes back into the 1800s, though, where people were recognizing that you could have vascular issues. It was thought originally vascular issues could lead to brain changes, but they really didn't have a way of really studying it back then.
The reason why I bring it up is because in popular culture, meaning TV, movies, the depiction of someone who has Alzheimer's is a complete dysfunction of the ability of the person to be a person. They almost revert to a childlike state. And I was hoping that you could confirm a theory that I have, at least when it comes to this, and that is the most late stage of the disorder, where there's almost nothing left of the original individual at that point, and that most people that contract Alzheimer's, it's much more subtler. And that's one of the reasons why I keep going back to On Golden Pond, because it's a very simplistic description of that kind of symptom in the easiest way that you can imagine it. It's getting lost in the woods that you've been at your entire life. Or like you said, it's a blocking of the normal and you can't figure it out. I think I've always characterized it as you know how to get from A to B, you know all the different ways,
Yes.
but you just can't make the connection between the two.
Yes, because it actually has been, that connection is either damaged
Mm-hmm.
or has been lost. And at first it's just damaged.
Yeah.
And it's very scary for the individual because they've done something a certain way
Mm-hmm.
all their life, and all of a sudden something has changed, and they've recognized something changed.
Yeah.
They can't explain it though.
Mhm.
But and that leads to that frightful type of situation,
Right.
of panic.
Mm-hmm.
But often that's how it starts. And then this is a disease that in some people takes 20, 25 years to progress.
Yep.
Other people it could be 5 years to progress.
Mhm.
It's a variable.
Well, I mean, that goes to exactly what we were saying earlier, that your homeostasis is not the same as mine.
Right.
I mean, take
Right.
the body temperature, for example. It's an average. Trying to explain to someone that some people run hot and that 99 degrees might actually be a normal temperature
Right.
for them, and it's not actually a fever.
Right.
Or someone can run at 94 degrees and they're not hypothermic.
Right.
So, yeah, the rate that we experience these things is variable, and disease is variable.
It is. And I think part of it goes to not only us being a genetic organisms that are a little bit different,
Mm-hmm.
but it's how we're exposed to our environment, and how our bodies can adapt easily or not.
Mm-hmm.
And that is the key.
Yep.
And if we, unfortunately, if we get exposed to the wrong elements, it's like anything. If you get exposed to, anthrax,
Right.
it could kill you.
Yep.
If you're not exposed to it, probably not.
Right.
Okay, so it goes to that issue. Okay, so it's in some ways it's luck of the draw
Yep.
of what we're exposed to, and what our genetics are, and how we can handle that.
Yeah.
Yeah.
Well, we're now at 6:31, and again, I don't want to keep you all night, even though we could.
Yes, I know.
And if I can, I would like for you to explain certain aspects of what you were just talking about with homeostasis a little bit more, if you could, in a little bit more detail.
Okay, with homeostasis, with the balance of
Yes.
Okay, well, homeostasis to me means that we are in balance with our nature, basically. We as an organism, so that we maintain a healthiness to us by maintaining this balance, and that means we don't have too many highs or lows in differences of how our bodies function. So that we're not overly stressing or under stressing the body. It's sort of like maintaining everything we do. So is sort of like that moderation issue.
Yeah.
Okay, if we consider it as moderation, what is that? So we're not excessive on anything that we're doing. We're trying to maintain the right amount. Now, what is the right amount for you, Joe, or for me, or
Right.
anybody else, any of our audience, is questionable. It's sort of like we find this empirically, meaning we discover it on our own. It's like, is drinking 2 cups of coffee for me the right amount? I think it is, but for somebody else, it may be excessive.
Oh no, I'll start having heart palpitations.
Right. So it comes down. It comes down to the individual and how you're living your life, and what you're doing in your life, what you're exposed to in your life, etc. That's where it is a bit of a crapshoot because the predictability of that is very, very troublesome.
No,
How do you
right.
predict? We can't predict these types of things. Some things you can predict. If for instance, if you're constantly doing something that is giving you, let's say you're drinking coffee, it's constantly giving you a heart palpitation. Well, you can predict that is potentially leading to AFib for you, right?
Potentially, yes.
Potentially. Okay, so you could predict that it's based on what you're doing. But if you're doing things where you have no recognizable consequence,
Correct.
you basically are still maintaining that homeostatic balance, because if you weren't, you would have some type of deficit.
You'd have a feedback.
Right, exactly. A feedback loop, etc.
Right.
Yeah.
And I think, because she put in here that she can drink 4 energy drinks back to back and be perfectly fine.
Yeah.
That actually might be caffeine tolerance that you're experiencing.
Yeah.
One of the best recommendations that I always give people when it comes to this is that you have to think of it like a microphone. And if you're drinking 4 energy drinks back to back, you're effectively moving that decibel augmentation down, so that even though it is something high volume, it's actually coming out low volume. If you were to spend 2 weeks without caffeine, which I know can be difficult in this day and age, and then have an energy drink, you would find that one has a much harder kick to it suddenly than it did before. But again, we can talk all night.
True.
So, Dr. Balin, I want to say thank you very much.
My pleasure.
Right now here at the university, we're kind of in a bit of flux. It's a little bit of a chaotic time because we're ending a block of stuff. So it's been a bit difficult for me to get new people on. But I do want to thank you very, very much for offering your time, sharing your time with us, and your knowledge.
Well, it's my pleasure. I'm learning things along the way as well. So I find this to be quite an enjoyable thing to do, and I'm happy to share whatever I have.
Well, thank you so much. As always, I would like to give you an opportunity to have the floor, say something that you'd like, ask a question that you'd like, anything.
Well, I want to thank the audience members that I don't see, but I know are here, and that could be here, listening to the YouTube recording as well, for actually having the interest and taking the time out to listen to us talk about these issues and to have great questions. Because that's what it's all about. Mm-hmm. We ask questions and we see problems in different ways, and we try to solve the problems. We try to understand biology. We try to understand what our world is about.
Perspective.
It's a perspective. That's correct.
Mm-hmm.
And the transference of knowledge. We all have different levels of knowledge. To me, that's really important, because not a single one of us has all of that knowledge. We have. It's disparate, right? It's based on who we are as individuals as well.
Yep.
So it's a learning process. I'm happy to share my knowledge with everyone here at the university and online as well.
Well,
so thank you.
Thank you again, Dr. Balin. I really do appreciate you being not just the fallback guy, but also our first researcher who was willing to step forward. And I still remember when I originally brought this up, and it was probably just like, oh, okay, well that sounds nice. But thank you for letting me run with it, and thank you again for being willing to join us here as the first one. Yeah. You're very groundbreaking in that regard. Planting your flag, I see.
Well, thank you, Joe, the science bro.
Well, thank you so much. Thank you very, very much. So allow me to just close us out, and then I can let everyone go back to whatever they were doing. So I want to thank you all for joining us tonight. I want to thank Aya. I want to thank Aru Warrior for spending a lot of time with us. I believe Casper was here at one point. So Casper, thank you very much for showing up. As always, guys, please remember to follow, as it helps the channel grow. Please remember to like, comment, and subscribe on YouTube. Also, please, if you can, share this playlist on YouTube with someone that you know, because it may actually give them some perspective, or it may actually help them learn something, like we're learning. We have advanced degrees, but yet, as I always say, you have to kind of look at everything with the eyes of a child. Everything has to kind of be new. So I want to thank you all for being here, and thank you all for giving us a chance to spend this time with you. So long, everybody, and thanks for all the fish. Bye-bye now.